Good afternoon. I'm extremely honored to have the opportunity to present my research to such a distinguished audience. Today, I'll be discussing my thesis project, which centers around unraveling alternative polyadenylation, or APA dynamics, from bulk RNA sequencing data. Through this investigation, I aim to shed light on the underlying molecular processes that contribute to the development and progression of various diseases. Now, genetic perturbations usually elicit changes in the levels of specific mRNA molecules. We generally assume that these mRNA levels are closely linked to subsequent protein levels. Although it is generally true that most mRNA molecules correspond to their respective proteins, there is a notable subset of mRNA that do not show a direct correlation. This inconsistency between the transcriptome and proteome indicates the presence of a post-transcriptional regulatory process. In other words, there are mechanisms that control protein production beyond the level of mRNA transcription. This dataset, along with numerous others, has revealed a noteworthy observation. The levels of mRNA and their corresponding proteins do not exhibit a consistent correlation. In this figure, we can observe the alterations in gene expression and their corresponding proteins in a Drosophila model tauopathy. The x-axis represents the log fold change of the transcriptome, while the y-axis represents the log fold change of the proteome. The orange dots in the figure represent genes where the levels of mRNA and proteins are in agreement, suggesting concordance. On the other hand, the blue and white dots represent genes where the levels of mRNA and proteins show disagreement, indicating discordance. As depicted in this slide, we have discovered that the majority of gene alterations do not align with their corresponding proteins as expected. Surprisingly, out of the 1,309 transcripts showing differential expression with accompanying proteome measurements, only 58% demonstrate consistent changes in the proteome. This discrepancy can be attributed to a range of post-transcriptional mechanisms, which present a considerable obstacle for gene set analyses. These analyses heavily rely on the assumption that transcript levels accurately represent protein levels. The lack of concordance between gene expression and protein expression poses a significant challenge when it comes to interpreting molecular data. The running hypothesis of our lab is that alternative polyadenylation, or APA, has the potential to bridge the gap between the transcriptome and the proteome, ultimately shedding new light onto the pathogenesis of various diseases. The APA machinery acts upon the pre-mRNA by cleaving it at either the proximal or distal cleavage and polyadenylation signal site, or CPAS site, within the 3' UTR. This process leads to the generation of different variants of mRNA with varying lengths of the 3' UTR. It's important to note that despite the variations in the 3' UTR length, both mRNA variants ultimately produce the exact same protein. However, the key difference lies in the lifespan of mRNA molecules. mRNA molecules with longer 3' UTR tails contain more binding sites for regulatory molecules such as microRNAs and RNA binding proteins, which typically results in a shorter lifespan for these mRNA molecules. This phenomenon of APA is widespread, as APA plays crucial roles in various biological processes, and its dysregulation has been implicated in several diseases, including neurodegenerative disorders and cancer. To study the dynamics of APA in various disease models, researchers have developed specific analysis methods that are designed for different sequencing protocols, such as deep 3' end sequencing and bulk RNA sequencing. This timeline provides a visual representation of the evolution of these methods in APA research, categorized based on the sequencing protocol used. However, despite their advancements, the current APA analysis methods have certain limitations. If we were to collapse all the issues from the current generation of bulk RNA sequencing-based APA methods, we encounter 4 major issues. The first issue is that they are hindered by a lack of accurate and comprehensive CPAS annotation system. These annotations fail to align with other CPAS databases, thus limiting the accuracy and reliability of our analyses. The second issue is that we encounter inherent limitations in the detection of de novo CPASs. The third issue we confront is the challenge of deconvoluting overlapping CPASs. Finally, the fourth and final issue revolves around the detection of intradistal or intraproximal APA changes. To tackle these critical concerns, we have developed a novel algorithm and methodology called PolyAMiner-Bulk. This approach not only tackles these aforementioned concerns, but also presents a comprehensive framework for analyzing APA changes using bulk RNA sequencing data. The three major advancements of PolyAMiner-Bulk consist of its unique CPAS-BERT deep learning model, the soft-clipped-assisted clustering mode, and the improved vector projection module as highlighted in this graphic. While there's a lot to unpack from each module, I want to specifically focus on our CPAS-BERT machine learning module and describe its significance in our PolyAMiner-Bulk methodology. So after generating our candidate CPAS library, we pass this dataset to our CPAS-BERT module, which can be compared to a sieve. In this analogy, CPAS-BERT acts as a sieve that effectively filters out artificial CPAS sites from our candidate CPAS library, ensuring that only CPASs with high confidence are retained. Now you might be wondering, how does CPAS-BERT actually work? To truly grasp the significance of CPAS-BERT in the realm of APA, it is essential to first understand the role of large language models like BERT in the context of the English language. I would like to first begin by presenting a simple sentence for our consideration. The graduate student went to the bank and learned of his empty account, after which he went to the riverbank and cried. Now, how can language models like BERT successfully differentiate between the word bank when it signifies a financial institution versus when it denotes the edge of a river? The key to understanding language lies in the self-attention mechanism employed by the model. This mechanism enables the model to grasp the connections between various words within a sentence. Simply put, the self-attention mechanism evaluates the significance or relevance of each word by comparing it with all other words in the sentence. This process involves calculating attention scores between the word being examined and every other word in the sentence. To illustrate, let's consider the word bank in our sentence. The word bank appears twice, but with different meanings in different contexts. When focusing on the first occurrence of bank, the self-attention mechanism assigns higher attention weights to the usage of bank that refers to a financial institution, as opposed to the meaning of bank referring to the side of a river. By taking into account the context and other words within the sentence, the model can discern the correct interpretation of bank in each instance. By utilizing the self-attention mechanism, the model effectively captures the relationships between words in a sentence and comprehends the context in which each word is employed. Now, the task of deciphering gene regulatory code to identify CPASs can similarly be modeled as a natural language processing task. The challenge of unraveling the gene regulatory code to identify CPASs can be approached by drawing parallels with natural language processing. This diagram illustrates that the APA field has previously discovered a specific region located 15 to 30 nucleotides before the CPAS, containing signal motifs such as the AAU, AAA. There are also regions situated 0 to 20 nucleotides after the CPAS, which contain GU or U-rich sequences. However, the order in which these motifs are arranged is crucial. Let's consider an example. If we were to interchange the GT or T-rich sequences with the CPAS signal motif, it would disrupt the fundamental CPAS grammar and render it incapable of functioning as a CPAS. Identifying CPASs is a highly complex task due to the presence of polysemy or multiple meanings and distinct connections in their semantic relationships. We have acknowledged that the long-standing challenge in the field of APA regarding the identification of CPASs. To address this, we propose a solution by fine-tuning the pre-trained DNA BERT model, specifically focusing on its BERT encoder architecture using task-specific data. In order to mitigate the occurrence of false positive CPAS artifacts, we have developed CPAS-BERT, an attention-based model. This model comprehends the unique semantic relationships within DNA sequences surrounding CPASs, thereby facilitating accurate and precise identification of CPASs. So in our training and testing data, we focused on positively labeled CPASs, which are candidates that both PolyACite and PolyaDB, two of the field's most widely used CPAS databases, agreed upon. As for the negatively labeled CPASs, we considered intergenic sites that are not located within 3 kb upstream or downstream of any annotated gene. This choice was made because approximately 99% of the human 3' UTR region is smaller than the 3 kilobases in length. So the performance of CPAS-BERT is exceptional, with accuracy, F1, precision, and recall all exceeding 0.9. However, what sets CPAS-BERT apart from other deep learning models is its unique advantage in interpretability. Unlike black box models, CPAS-BERT is built on the transformers architecture, which provides a highly interpretable framework. Therefore, we have employed a visualization technique to enhance the interpretability of CPAS-BERT by highlighting important regions in positively labeled or CPAS-containing sequences. The attention landscape on the left clearly illustrates that CPAS-BERT exhibits heightened attention in the expected regions, namely the 15 to 30 nucleotide region upstream from the CPAS and the 0 to 20 nucleotide region downstream from the CPAS. Furthermore, we conducted motif enrichment analysis on these highly enriched regions and successfully identified well-conserved CPAS upstream and downstream signals, instilling confidence in CPAS-BERT's ability to learn the essential features required for accurate CPAS identification. This data highlights the strength of attention-based deep learning models, as they surpass the simplistic assessment of motifs' presence or absence. Instead, these models leverage contextual understandings to simultaneously comprehend multiple semantic features. So during my PhD journey, I've been fortunate to employ our PolyAMiner-Bulk platform to investigate APA in various disease models. I'm profoundly appreciative of these collaborations with these esteemed labs and researchers who have significantly enriched my understanding of APA and its implications in diseases like glaucoma, meningiomas, heart failure, scleroderma, pancreatic adenocarcinoma, and glioblastomas. These projects are at various stages of the publication cycle, but I encourage you all to explore APA in your own existing bulk RNA sequencing data using PolyAMiner-Bulk. That being said, this concludes my presentation. I would like to take this moment to express my deepest gratitude to these incredible individuals and institutions who have played an instrumental role in making this research possible. Thank you.
Thank you, Dr. Donacudi.